Hello everybody, in this video lecture we will cover chapter 12, Diversity of Life. So all life on Earth evolved from a common ancestor. Biologists map how organisms are related by constructing phylogenetic trees. So on a diagram on a diagram below, you can see example of this phylogenetic tree. So all these branches represent different groups of living organisms, and you can see they are all connected, and those points of connection show us common ancestry. So if you go deep enough in time, we come to our LUCA, or last universal common ancestor, and this diagram shows us that all life on Earth are related from this organism. So systematics is part of biology, it's a branch of biology, and systematics focuses on classifying organisms and determining their evolutionary relationship. So taxonomy is part of systematics, and taxonomy is the identification, naming, and classification of species. So systematics include taxonomy. Right, so you can see we have systematics here that will include taxonomy, classification, nomenclature, or naming, and morphology. So some basics of taxonomy. Carlos Lunier proposed the current taxonomic system based on a two-part name for each species and hierarchical classification of species into broader groups of organisms. So that's the guy. So each species is assigned a two-part Latinized name or binomial, consisting of genus and name unique for each species. For example, for humans, our scientific name is Homo sapiens. So you can see that Homo means our genus, that's a bigger group, right? And Homo sapiens is our unique name for our species. So if you look how it's written, you can see that it's a two-part name and it's in italics. It's giving Latin ending, and the first letter of the genus is capitalized. So that's the right way to write a name of species. So species that are closely related are placed into same genus, right? So if you look at these big cats, right, you can see their common names like leopard, lion, tiger, jaguar, and in the parentheses you can see their scientific names for their species, and it's always two words, genus and species, with the first letter of genus that is capital. And you can see they are different species, but the genus Panthera is the same, and it tells us that they are closely related group of animals. And so if you look over here, you see the leopard and scientific name, right? So that's a species name. The first name is genus. And then we progress to a broader categories of classification. So from genus, we go to family, order, class, phylum, kingdom, and domain. So domain is the bigger group, right? Kingdom is smaller than domain, and phylum is smaller than kingdom. Classification of eukaryotic diversity have changed rapidly in the recent years. The 4 eukaryotic groups within the 5 kingdom classification, plants, animals, fungi, and protists, have been transformed into the current system of 6 supergroups. So before, when I went to college, right, we used 5 kingdom classification. So all life was classified as plants, animals, fungi, or protists, right? And then you see it's 1, 2, 3, 4. And then it also was, well, way back it was Monera, but then they divided it into bacteria and archaea. But anyway, we used to have these kingdoms and we don't have them anymore. We substitute them with supergroups. The intent of the supergroup classification system is to unite microbial and macroscopic eukaryotes based on phylogenetic inference. So let's look on the next slide on these supergroups. So here we have life. Life is divided into 3 domains: domain bacteria, archaea, and eukarya. And domain bacteria has supergroup bacteria, domain archaea has supergroup archaea, but eukaryotes are divided into these 6 supergroups. Now, I want to just briefly remind you that bacteria and archaea, they are prokaryotes, and prokaryote means they don't have nucleus. They do have DNA, however. Eukaryotes, those are organisms whose cells have nucleus, and inside this nucleus we have most of the DNA. Right, so that domain eukaryota is divided into excavata, amoebozoa, apistaconta, rhizaria, chromalveolata, and archaeoplastida. And if you see over here that excavata, for example, includes protists, right? It's specifically protozoa, flagellated protozoa. An example would be Euglena. Amoebozoa include mostly amoebas, right, and slime molds. Apistaconta over here includes animals, fungi, and this organism that's also protist. So pretty much we have this protist, and protist used to be its own kingdom, very, very simple living organism. They were not animals, not plants, not fungi, right? They were plant-like, animal-like, fungi-like, right? So that was, microscopic organism. And now they put them within a group with more complex organisms to show that they are really related, right? So here we have a relationship between animals, fungi, and choanoflagellates that is protist. So over here we have brown algae, diatoms. In Archaeoplastida we have land plants, green algae, and red algae. And green and red algae also used to be in the kingdom Protista. So it's very interesting classification. It's way more complex, especially for students like you guys who do not major in biology. But life is complicated. We cannot simply divide everything into plants, animals, fungi, right? That's our supergroups. We will briefly look at different supergroups. So bacteria. Bacteria are prokaryotes. The 3 most common shape of bacteria are spherical, cocci, rod-shaped, bacilli, and spiral or curved. So here you can see cocci, bacilli, and spirals. Archaea is another supergroup, and they also prokaryotes, but very different from true bacteria. They live in extreme environment. So this is Yellowstone Park, and they have like hot water there, like a lot of sulfur, and this is a good area for archaea to live. And what archaea does, it just create really incredible ecosystems over here. The colors are just so unique. If you ever go to Yellowstone Park, you will feel like you're on a different planet. So everybody should go to Yellowstone Park. That's really a miracle on Earth. Excavata, another supergroup, an example is Euglena. So you can see Euglena is protist. It's a small unicellular organism, And it's something in between plant and animal. It does has chloroplast, so it can do photosynthesis like plants do. At the same time, it has a flagellum and it has some sensory area. It's like an eye of an animal, and they can move towards light. So movement is the characteristic of animals, and photosynthesis is characteristic of plants. So here we have living organism that is in between. It's a plant and an animal in one living microscopic organism. That's really amazing that we have organism like Euglena. Amoebozoa includes some example amoeba or slime molds, so very closely related organism over here. And amoeba is also unicellular organism living in the water, in fresh water. Apistoconta. So Apistoconta includes animals and fungi, right? So here you can see animals, well, some mammals, right? So like all different examples. Fungus, starting with some mushroom. Some of them are poison, and over here that's a athlete feet. So that's parasitic fungi. Rhizaria example would be foraminifera. So foraminifera are microscopic living organisms. They look just very, very beautiful, right? You cannot see them really with the naked eye. Some of them, foraminifera, might be a little bit bigger, but under microscope, that's how they look like. Chromalveolata include brown algae, and this is a kelp, common in our area of California, right? So it's also used to be classified just as a protist. Archaeoplastida includes green algae and land plants. So here you can see different example of green algae. Diversity is just amazing. And here land plants. Okay, so that very, very brief introduction. It's nothing to talk about any, deep knowledge, just to show you how incredibly how incredible life on this planet is and how diverse it is, how many groups, so different and so unique sometimes, and at the same time we know that they all are related through this common ancestor, right, through our Luca. Now, we already mentioned that life is divided into three domains: bacteria, archaea, and eukarya. And eukarya includes what we think about life, right, like plants, animals, fungi, so on. So eukarya are large, more complex cells, each with a nucleus inside and membrane, inside a membrane and other membrane-bound structures. Archaea are tiny, tough prokaryotes with no nucleus. Many are extremophiles, so they live in very harsh conditions, in harsh environment, like too hot, maybe too salty, maybe too, lots of sulfur, and so on. Bacteria are true bacteria, prokaryotes with no nucleus, found pretty much everywhere where life can possibly exist. Here just give us some classification of three different organisms like chimpanzee, us, human, right, and baboon. And you can see how we can put it into kingdom, right, phylum, class, order, family, genus, and then species. So we all are different species, right, and I'm reminding you that means we do not breed, and chimpanzee does not breed with baboon. Right, and when we look at the genus, you can see that we all in a different genus, right? Because that's what make us, the species include genus and species, and then we are in the same family with chimpanzee, and we are in a different family with baboon, right? And that means that we are more closely related to chimpanzee than we related to baboon, right? So that classification also allows us to see the evolutionary relationship between different species. Now, cladistic is the part of systematics when organisms are grouped by common ancestry. Clade consists of ancestral species and all its evolutionary descendants and forms a distinct branch in a tree of life. So cladogram is a diagram which shows relationship among organisms. So all cladograms are diagrams with lines, right? And where these lines intersect, that's where this intersection shows us common ancestor. So how to build a cladogram? To build a cladogram, first we need to make this table, and in this table we're looking at ancestral traits and derived traits. So ancestral traits, it's a trait that we share with a common ancestor, and derived traits are newly evolved traits. So if you see over here, we have lamprey, shark, salamander, lizard, right, tiger, gorilla, and human. And you can see that we can look at these different traits, jaws, lungs, so on. And a lamprey doesn't have any of it, but shark has a jaws. So jaws is a new derived trait for a shark, but for salamander, jaws is already ancestral trait. So salamander inherited it from the ancestor that they shared with the shark. And then something new appears, right? So lizard now has two ancestral traits and some derived, and so on. So the point is the speciation and evolution is remodeling. You remodel, you add something, you remove something, but you never really start with a scratch. So let's say gorilla was not formed from completely something new. It was not started with a scratch. Gorilla has lots of characteristics that it shares with tiger, lizard, salamander, shark, and lamprey, and so on, right? So that was a process of adding and removing something. And that's what evolution tells us. It's a remodeling process. It's not creation of a new, completely new organism with very specific characteristics. And after we make this table, we put it on a diagram, and that's a very simple example of a cladogram over here. So, well, you can pause the video and try to do this cladogram and see if you can arrange this living organism on this diagram. So now how to read cladograms? So if you see cladogram over here, we can notice that koala and kangaroo, right? So here's koala and kangaroo. They are more closely related to one another than to anything else on the cladogram. Because kangaroo and koala, their lines intersect first. That means here they have common ancestry. And remember, time goes up. So that's at this point of time, it was an organism that then evolved into two different groups. And, to have a common ancestor, let's say with a bat, we need to go deep in time, right, over here. So this indicates that they derived from each other more recently. Diverged. I apologize. This indicates that they diverged from each other more recently than they diverged from any of the other animals on the diagram. Okay, so over here, if we look in, let's say, shark and tuna and a human, right? So let's look at the three animals, shark, tuna, and human. And the question would be, is tuna more closely related to human or to a shark? Are we derived do we share more recent common ancestor with tuna, or tuna shows more recent common ancestor with shark, right? Um, so again, you're looking where line intersect. So tuna and human, our line intersects first, early in time, then in shark, tuna and shark. So tuna is more closely related to humans than it is to shark. That's really confusing over here, right? But what you think, you just see what lines intersect first, right? That's a common ancestor over here. So phylogenetic trees are hypotheses about evolutionary history. Lumière's divided all known forms of life between the plants and animal kingdoms. So we used to have only two kingdom system, right? Plants and animals. We didn't even know that other form of life exists. And this system was prevailed in biology for over 200 years. In the late 1900s, molecular studies and cladistics led to the development of a three-domain system, recognizing two domains of prokaryotes, bacteria and archaea, and one domain of eukarya. A new classification was produced in 2005 for the International Society of Protistologists. So that's, people who study those protists. And it divided eukaryotes into 6 supergroups that we discussed earlier. The published classification deliberately did not use ranks like kingdom. So they didn't want to use kingdom to show that that was wrong. As of December 2010, there appears to be a consensus that in 2005, 6 supergroup model does not reflect the true phylogeny of eukaryotes, and hence how they should be classified, although there is no agreement as to the model which should replace it. So why would I, finish with a slide like this? Because I want to tell you that classification is a work in progress. We getting new information, we performing new analysis, we analyze DNA, molecular data, and we, correcting some maybe, mistakes, or even if it's not mistakes, but just some knowledge that we had before that now does not, knowledge that need to be changed because of the new research, new data, new analysis, right? So we're working on it. Classification is something that will be changed in the near future, probably again, but that's okay. That's good for science, because science is never arrogant. Science is not about I was right and that person was wrong. Science about trying to find the truth, right, and use this truth to serve the society. So that's it for this chapter, and I hope it was helpful.